Topomerase is the first enzyme in DNA replication. It unwinds the DNA from the double helix structure to this single strand right here. This sets up the strand that is replicated into two DNA strands. The next enzyme is helicase. Helicase breaks the hydrogen bonds between nitrogenous bases to separate the original strand into two anti-parallel strands, like this. This creates one strand that runs in the 3' to 5' direction and one strand that runs from the 5' to 3' direction. During the DNA replication process, this part where helicase breaks the hydrogen bonds is called the replication fork, and over here where the new DNA strands are being built is called the replication bubble.
The single-stranded binding proteins bind to the individual anti-parallel DNA strands and keep them apart after they are separated by helicase.
Next, what happens is primase will add a primer. The primer is added next to the origin of replication so that the DNA can build in the 5' to 3' direction. The lagging strand of primase is added near the helicase so that the DNA strand can also build from the 5' to 3' direction. This is a leading strand because as helicase continues to break more H bonds, DNA polymerase can continue to build in the 5' to 3' direction for the new strand of DNA as it is building off of this primer right here. And then down here is a lagging strand because as helicase continues to break more H bonds, this primer is pushed back. A new primer is going to be built right here following this helicase because DNA polymerase can only add nucleotides after a primer. DNA polymerase for the leading strands adds nucleotides up here, one at a time, heading towards the helicase so that again the DNA can build in the 5' to 3' direction.
Bro, what are you looking at?
And then for the lagging strand, DNA polymerase will build off of the primer, heading in the 5' to 3' direction. And then again, as stated, as helicase continues to break more H bonds, more primers will be needed down here on the lagging strand for DNA polymerase to continue to add nucleotides.
DNA polymerase 1 swaps out the RNA in the primer and adds in new DNA.
We believe we have more for the helicase. No, I'll do video.
We mentioned maybe like this is the leading strand.
Where is the origin of replication?
And then DNA polymerase 1 will also take out, or it'll check for any errors in the DNA and it'll replace them.
So where's the origin?
The DNA ligase is an enzyme that glues together the Okazaki fragments by phosphodiester bonds, creating a fully continuous strand. And then after the new DNA is created, the telomerase are the repetitive bases that are non-coding on ends created by telomerase. They are only in stem and cancer cells.
The original DNA replicates so that it has one strand of the newly created DNA and then one strand of the original.